So good afternoon, everyone. Thank you for joining us virtually for today's seminar. It's with great pleasure that I introduce today's phenomenal speaker, Dr. Stephen Luby. Dr. Luby is a world-renowned scholar whose research and work has made profound impacts on the public health community for decades. Dr. Luby earned his bachelor's in philosophy summa cum laude from Creighton University. He then earned his medical degree from UT Southwestern at Dallas and completed his residency in internal medicine at University of Rochester Strong Memorial Hospital, followed by fellowships at the CDC, including the Epidemic Intelligence Service and Preventative Medicine Fellowship. Following this, he held positions at Aga Khan University in Pakistan as assistant professor in epidemiology, the foodborne and diarrheal disease branch at CDC as a medical epidemiologist, and the International Center for Diarrheal Disease Research, Bangladesh, as the director, before joining Stanford, where he is a professor of medicine, senior fellow at the Woods Institute for the Environment and at the Freeman Spoyle Institute for International Studies, associate dean for global health research, and professor by courtesy of epidemiology and population health. His research centers around advancing solutions to a broad array of public health problems, including reducing air pollution from brick manufacturing in Bangladesh, reducing exposure to lead, advancing vaccine evaluations for typhoid and Nipah virus, and providing safe water to low-income communities while preserving the environment, just to name a few. We eagerly anticipate his enlightening presentation and the valuable insights he will share with us today. Without further ado, please join me in extending a warm welcome to Dr. Stephen Luby.
Well, thank you very much, Steph, for those kind words. As part of this conversation, I was asked also to talk a little bit about my trajectory. So just to kick off, as Steph noted, I am a physician, and I actually went to medical school intending to work as a public health professional, reducing the burden on disease in low-income countries. I did not originally intend on a career in research, nor did I plan to work in emerging infections. But I'll talk about more my pathway as I discuss today's topic. When we think about what we know about Nipah, it really flows from a remarkable collaboration of many, many people across many geographies and many institutions. And I don't have the time to call them all out at every step, but I just want to begin by noting that the synthesis of this work really comes from a whole community working on these questions. So Nipah virus first came onto the radar with a large outbreak in Malaysia. Between September of 1998 and May of 1999, 283 human cases of acute encephalitis were identified, with 109 deaths. That's a case fatality rate of 39%. Paul Schwal, he isolated a novel paramyxovirus from a patient in Kampong Sungai, Nipah village, hence the name Nipah virus. If you look at the clinical features of the people who were infected, some of them had moderate symptoms: high fever, headache, muscle aches, vomiting, drowsiness, or dizziness, symptoms that are typical of other viral infections. But some of these folks progressed to more severe disease, where they got encephalitis. So encephalitis is inflammation in the brain. And with this encephalitis, they had symptoms of disorientation, hallucinations, seizures, convulsions. Some of them progressed to coma and death. So an immediate question was, how did people contract this novel virus in Malaysia? Well, the outbreak was concentrated among pig farmers. Malaysia is a predominantly Muslim country, so pig farmers are a minority. So pig raisers were a distinct minority. 92% of cases reported contact with pigs. And when a case control study was conducted, compared to controls, people with Nipah encephalitis were 5.6 times more likely to have close contact with pigs and 3.7 times more likely to have contact with sick pigs. So this virus which was originally identified in people, spilled over from pigs. So, or was transmitted from pigs. This was a zoonotic disease, a disease from animals to people. But people have been raising pigs for a long time, and we hadn't seen Nipah before, so it suggested that it was likely not the reservoir host, but rather was an intermediate host. So investigations were done to figure out where this virus was coming from. That is, where did the pigs get Nipah? So numerous wild animals were trapped, and 4 out of 8 species of fruit bats had antibodies against Nipah. And one of these guys, a Pteropus hypomelanus, in Malaysia, a virus was isolated from urine under a roost of Pteropus hypomelanus bats. So the bats were thought to be the reservoir species. They don't get sick when they're infected. But if you look at Nipah virus and what we've learned about this, it doesn't just stay in bats or in pigs or infect people. It is a generalist. It has broad species tropism. Of course, we're very familiar with SARS-CoV-2. It's also a generalist virus that can go into lots of people, lots of organisms. Nipah virus is able to infect 6 mammalian orders. It's just incredible. So Nipah enters mammalian cells through the ephrin B2 or ephrin B3 surface glycoprotein. That's the surface receptor that the virus comes in on. And I took notes through all of medical school, but I don't remember the ephrin B2 or B3 receptor ever being mentioned. It turns out, though, it's quite important. It's critical for growth, maturation, and remodeling. It's part of the cell-to-cell signaling that allows vascular, nervous, and skeletal cells to develop and to communicate with each other. It is such a crucial molecule that if you have a pet dog or a pet cat or a pet hamster, their ephrin B2 and B3 is nearly identical to your own. 95 to 98% DNA sequence homology of ephrin B2 or B3 across mammals. And that's why this virus can get into almost any mammal, because we all have these ephrin B2 and B3 surface glycoproteins. So the index farm in Malaysia was a large farm that had 30,000 pigs. This is a picture of the farm, and you can see it's adjacent to primary forest, which was fruit bat habitat. And there was a network of other farms close by. They actually moved pigs from one farm to another, depending upon where they were in their development cycle. So what was thought to have happened is it became increasingly common among pig raisers in Malaysia to plant mango trees around the pig enclosures. And they did this because, well, who doesn't like mangoes? And you could sell the mangoes as another cash crop. And if some of the mangoes fell into the pig enclosures, well, the pigs would eat them. The problem was that the mango trees were apparently thought to attract fruit bats. And fruit bats are messy eaters. They'll eat and they'll spit, and they'll eat and they'll spit. And anything that falls down into the pig enclosures, well, the pigs will eat. They are, after all, pigs. So the virus was thought to go from the respiratory secretion of bats to pigs. As pigs got infected, they had a primarily respiratory illness, and that this then was transmitted to the people who cared for the pigs. So Malaysia, the government of Malaysia undertook a large culling operation where they killed over 900,000 pigs. They didn't allow fruit trees to be permitted around pig enclosures anymore. The pork industry was decimated. It still hasn't recovered 20 years later, 25 years later. but no subsequent cases of Nipah have been reported from Malaysia, either in people nor in animals. So the global health community had some takeaways from the Nipah outbreak in Malaysia. First, Nipah was a newly recognized high mortality pathogen. It was a zoonotic disease that moved from animals to people. The primary disease was encephalitis, and person-to-person transmission was not noted in the Malaysia outbreak. I recall attending a lecture on Nipah when the team who had investigated with Malaysian collaborators returned and reported on their findings. I was working in the foodborne and diarrheal diseases branch. I recall thinking, very bad disease. Glad I'm not working on that one. And then went back to work on more diarrhea. But in August of 2004, I moved to Bangladesh. I was appointed head of agency for the U.S. Centers for Disease Control in Bangladesh, and the focus here was a risk on emerging infections. The CDC was particularly worried about Bangladesh because of the very high population density. Bangladesh has the equivalent population of all of the U.S. east of the Mississippi, all living in a country that is the size of the state of Georgia. So it is a very densely populated country, and people have very close contact with the environment. So with my position, I prioritized collaboration with the government of Bangladesh, working on surveillance and outbreak investigation. So 5 months after I arrived, in January of 2005, government health workers reported that 8 previously healthy people from Bashail subdistrict died within a 1-week period. 3 days later, the government of Bangladesh invited ICDDRB, that's an international research institute that I was seconded into. They invited us to collaborate with them on the investigation. So when we went up to Bashail Upazila, we found not 8 patients, but ultimately 12 case patients that met our case definition. All of them had fever. 3/4 were unconscious. Most concerning, 11 of the 12, or 92%, died. And the median time from the first symptom to death was 4 days. So at that point, we did not know what we were dealing with, but we knew it was serious. So being epidemiologists, we begin with person and then move to place. When you look at the map of where these cases occurred, you can see that there are 2 clusters. There's a cluster right there by the road, another cluster in another spot, and then 2 others that are a little farther away. But it's really striking that the cases were clustered in space. And when we look at the outbreak based on time, we also see clustering in time. The entire outbreak occurred over the course of 17 days, with most of the cases, all but 2, occurring over 7 days. So this was an outbreak that clustered in both space and time. And by the time we got there, most of the outbreak was over. We were able to collect specimens from 3 people in the outbreak, the first case, the sole survivor, and the other 2 cases that occurred later. At that point, we didn't have diagnostic capacity yet set up in Bangladesh, so we sent the specimens to CDC in Atlanta, and it took a couple months. But ultimately, what we found was that 2 of the cases had positive antibody, both IgM indicating acute infection and IgG suggesting that an even longer-term antibody developed. One of the cases in the outbreak had a negative antibody response, but this was something that was also observed in Malaysia. One of the issues is that people die so quickly from Nipah that they sometimes don't even have the time to mount an antibody response. So we were confident that the serological data confirmed that the outbreak in Tongyle district was Nipah virus. So there had been some studies done in the Indian fruit bat. Pteropus medius is the only member of the Pteropus genus that lives in Bangladesh. And whenever these bats have been studied, a solid proportion of them have antibodies against Nipah virus. So the question in Bangladesh was, how does Nipah virus, how does this bat virus transmit from wildlife to humans? So this kind of situation, I would argue, is really standard in epidemiology, that we have a disease and we're wondering what exposure leads to that disease. So our study question was, what exposures were more common among people with disease compared to people without disease? So we did a case control study. The cases were all 12 persons who met the case definition. And controls, we took 3 controls per case. We went to the next closest household. We took the person nearest in age, and then went again to the next household to get 3 of them. Because 11 of the 12 cases had died, we had to ask friends and family about the exposures of the people who were cases. So this is what we found. We begin with looking at the proportion of exposures among cases. So one of our leading hypotheses was that cases climbed trees and in the trees got in contact with bat urine or bat saliva. It was striking when we went up to the village, we saw lots of kids climbing trees. And sure enough, 25% of cases had climbed trees, but so had 31% of controls. So obviously there was no difference between cases and controls. And that was the same thing when we started to look at other exposures. There were no pigs in the area and no pigs had come through. We asked about other animals and there was no difference in exposure for animals among cases and controls. Similarly, we looked at fruits. These are fruit bats, after all, and we found no difference in fruit consumption among cases or controls. We looked at whether people had traveled, whether they had contact with sick people, and again, no difference. We kept looking and looking. I'm just showing you a summary. We had pages and pages of exposures that we looked for, but it turned out that only one of them, that is drinking raw date palm sap, was different between cases and controls. Cases were 7 times more likely to report drinking raw date palm sap than controls. So the next question that raised is, what is date palm sap and why would anybody be drinking it? So we did an anthropological investigation to understand that practice. And date palm sap is a little bit like maple syrup in the northeastern United States. In the wintertime, in late November through March in Bangladesh, sap harvesters cut a tap into the tree, and in the evening, they place a clay pot under the tap, and they remove the pot in the morning. Now, most sap is boiled into molasses, but some is sold fresh early in the morning. This is a local delicacy. And some villagers reported bats visiting their trees at night. The son of a date palm sap collector died in the outbreak. This young boy drank fresh date palm sap daily. He loved it. Several days prior to the outbreak, the collector sent date palm sap to his relatives in a nearby homestead, and 3 cases occurred in that family. That was one of the clusters that was on the map. So the conclusions we drew from the Tongyle outbreak investigation was that Pteropus bats occasionally shed virus into the sap, and when people drank the sap fresh, they became infected. But both the Tongyle investigation and other investigations in Bangladesh demonstrated that Nipah presented differently in Bangladesh than it did in the defining outbreak in Malaysia. In Bangladesh, we didn't see intermediate animal hosts. respiratory symptoms were very common. Over 2/3 of patients' cases developed respiratory difficulties. And there was also a strikingly higher case fatality rate, 75%. Now, the medical facilities in Malaysia were better than in Bangladesh, and many patients went on respirators, ventilators to survive, but there was at least a crude case fatality that was quite a bit higher in Bangladesh. Following up on the identification of date palm sap in Tongile, we continued to mount outbreak investigations that we identified through our surveillance system. And in multiple subsequent outbreaks, we again found strong statistical associations between reported consumption of date palm sap and Nipah virus. So part of what we wanted to do was to develop an intervention to protect people. And this ultimately evolved over years into a two-stage intervention. And I don't have time in this seminar to go through all of the steps in developing this. But what we worked on was first communicating to people the risk of drinking raw date palm sap. So we said, if you want to be protected, the safest thing to do is to avoid drinking raw date palm sap. And we told people this, and the government told people this, but they by and large ignored us. They continued to enjoy drinking raw date palm sap, and they didn't really believe it was unsafe because they and their family members had been drinking raw date palm sap for centuries, or at least for generations, as long as they could remember. And their belief was that this disease was caused by a divine curse. On the other hand, we said, well, we did try some harm reduction. That is, if you're going to drink sap, drink only Bana protected sap. So Bana is the Bengali word for skirt. And so we encourage making bamboo skirts that would keep the bats out of the date palm sap. This provided some benefit to the date palm sap collectors because that way the bats didn't follow the sap. And if people saw that the skirts were being used, they could be confident that the sap was bat protected. So this is all focused on this issue of how do we deal with the virus spilling over from bats to people. But there's another dimension of Nipah that was also concerning. And again, I want to tell the story through an outbreak. So the first case in the Faridpur outbreak had onset of symptoms on February 19th, 2004. He was from a village in Malikpur, and he enjoyed drinking raw date palm sap. He got seriously ill. Now, when people in Bangladesh get ill, they turn to their family to really give them care. Even if they get hospitalized, it will be family members who provide most of the hands-on care. And this family care is really rooted in emotional support and is part of the culture. There's a cultural expectation to maintain close physical contact during illness, to provide hands-on care with direct contact with the patient's body fluids. There's a real desire to have close physical contact before the person dies. People want to hug to say goodbye. They want to try to feed the sick patient or whisper Quranic verses in their ears. Family members, religious leaders prepare and cleanse the body, particularly the orifices, for burial. So patient A's mother, son, friend, and aunt cared for him during his fatal illness. And 2 weeks later, those 4 caretakers developed a similar illness. Now, case B was the index case's aunt. And she was a member of of an unusual religious sect that was actually based in a nearby village. And so when she became very ill, the leader of the sect came to visit her and to minister to her while she was dying. And a couple of weeks later, this religious leader developed a severe illness. And when he became sick, this was really a crisis for this entire religious community. He really was the leader of it. And because of that, many people came in to be with him as he was sick and dying. And a couple of weeks after that, many of those who cared for him became ill, and many of those who cared for those became ill. So, over the course of 5 generations, we saw 36 cases of Nipah infection in Faridpur. 23 had antibody Nipah virus confirmed. 13 had insufficient samples for testing. And once again, we see 75% of them died. So this was a clear illustration of human-to-human transmission of Nipah virus. And this, in fact, accounts for over half of Nipah cases that are recognized in Bangladesh. But human-to-human transmission is relatively inefficient. This is not like the Omicron version of SARS-CoV-2. The R0 for Nipah in Bangladesh, as we look over like 20 years worth of data, averages to be 0.3. So R0, or the reproductive number, is the average number of people that one sick person will infect. So if there's one person, on average, they're not going to infect anybody. But you could imagine if there are 10 people that would be infected, then 3 of them might transmit on, and of those, then we might go to another generation. But Nipah tends to cause these local high mortality outbreaks, but the chains of transmission are not sustained. Nevertheless, as communicable disease epidemiologists worried about emerging infections, we try to better understand these transmission events. So if we think about viral transmission, on the one hand, there's a risk of transmitting, and on the receiving end, there's a risk of becoming infected. So let's turn first to the risk of transmitting. Who transmits Nipah? Well, it's overwhelmingly people who die. 11% of all people who died in our long-running surveillance in Bangladesh transmitted Nipah to one of their care providers. None of the survivors transmitted. The people who transmitted tended to be older and tended to be patients with difficulty breathing. So the confidence limits are wide here, but if you look at the estimated reproductive number among people who are over the age of 45 with breathing difficulties, the R0 approaches 1. If we then look at what are the risks of becoming infected, and this is a cohort study undertaken during the Faridpur Nipah outbreak. It's touching a Nipah patient who later died increased your relative risk by 15 compared to people living in the village who didn't touch a Nipah patient who later died. And if you look at other characteristics or other exposures, they're all, again, related to this physical contact with an unconscious patient or touching a patient with respiratory symptoms. Notably, people who washed hands after contact with patient F, that is the religious leader, were 80% less likely to develop Nipah compared with those who contacted patient F and didn't wash their hands. So why do we see person-to-person transmission in India and Bangladesh, but not in Malaysia? Well, I think that there are 4 major hypotheses, and I don't think any one of them is correct. I think they're probably all contribute. Looking back at the data, it looks to me that transmission that was person-to-person did occur in Malaysia, but was unrecognized at the time. Caregivers in Bangladesh are likely exposed to more virus because of the way they interact with people who are sick. And it also looks to us that people in Bangladesh get a higher dose of, received a higher dose of virus by drinking date palm sap compared to being exposed to respiratory secretions from pigs. We're also particularly concerned that there may be a genetic difference between Bangladesh and Malaysian strains because of the impact that might have as we think about future risks. So if you look at this is just looking at the phylogenetics of the Nipah N gene, and you can see that there is some difference between the Malaysian strains and the Bangladesh strains. And Michael Lowe and colleagues looking at this estimate that the Nipah Bangladesh and Nipah Malaysia strains diverged from each other about 75 years ago. So this suggests that the virus continues to change. It's not sitting there immutably. It's not changing as fast as HIV. It's not changing as fast as SARS-CoV-2, but it is changing. And so we wondered about how these different strains of Nipah virus might behave in animal models. So Brown and Clayton took 8 ferrets and inoculated them with the Nipah strain, with a Nipah strain from Bangladesh, and 7 ferrets she inoculated with the Nipah strain from Malaysia. Then looking at the mean viral RNA levels in respiratory secretions, you can see that the Bangladesh strain had a lot more RNA copies, 10 times higher among ferrets inoculated with the Bangladesh strain compared to ferrets isolated, to ferrets infected with the Malaysian strain. So these data suggest genetic differences affect transmissibility. So if we think about sort of the risk of emerging infections, we worry a lot about zoonotic pathogens. So zoonotic pathogens spill over into humans, but usually they don't become human-adapted pathogens. If a zoonotic pathogen increases its capacity for person-to-person transmission with an R0 greater than 1, then a new pandemic can emerge. Each time a person is infected, the pathogen is in an environment which selects for adaptation to human transmission. So Nipah becomes then not just a local problem, but a global pandemic concern. RNA viruses have the highest rate of mutation of any virus or living organism. Nipah virus infects dozens of humans each year, and each human infection provides an environment that selects for emergence of strains with improved transmission. Advances in synthetic biology are progressively reducing the barriers to weaponizing high mortality pathogens. So emergence, natural emergence, or the engineered development of a more transmissible strain would be a catastrophic global pandemic. We saw what a disease that initially had maybe a 2% case fatality rate with SARS-CoV-2 did to the globe. It's kind of unimaginable, but to think about what a 75% case fatality pathogen would have on humanity. In addition, even the people who survive typically have marked mental deficits. So as we think about this threat, the threat that we might have a devastating Nipah pandemic, it raises a broader question about which future threats should we allocate resources to address. So one response to the concern that would be that we shouldn't focus on low probability scenarios. We should instead focus on things that are more likely to happen and not waste time on low probability outcomes. On the other hand, if a Nipah outbreak occurred, it would be really bad. So maybe even though the probability is low, maybe we should invest. So we might think about this in 4 quadrants. So it's possible that there might be no new celebrity gossip available today in the mass media or on social media. But I think that that's a really low probability, but it also has low impact, so we don't need to worry about it. I would argue it's a higher probability that the Atlanta Hawks are going to lose some games in March. I apologize to the Hawks fans, but I argue this is high probability, but again, low impact, so we don't need to worry about it. On the other hand, if we think about worsening climate change, this is high probability, it is coming, and it is a high impact concern. So we know that we should be spending resources, investing to reduce the consequences of climate change. Nipah pandemic is a low probability but high impact event. And I would argue that prudent actions should be guided both by probability and impact. So I'll give some examples. If you are a parent of young children, many of you will buy life insurance. That's not because you think you're going to drop dead, but in the low probability that you do, the impact would be so terrible if your children were not able to be supported financially that you buy insurance. Or we might even think about with seatbelts. We don't put on seatbelts. You don't put on a seatbelt when you get in a car because you think you're going to be in a high velocity collision. Rather, you just accept this investment of the time and the habit to put on a seatbelt in the low probability scenario that you would actually have a high impact collision. So my research group continued to work on Nipah, and we published 30 manuscripts on Nipah virus between 2006 and 2016. These demonstrated recurring spillovers, high mortality, and person-to-person transmission. Then the editor of Antiviral Research asked me if I would be willing to put an article together on the pandemic potential of Nipah virus. I'm not one who likes to write a lot of commentaries. I prefer to have to make arguments based on data, but I felt that this was an issue that would benefit from some focused attention because there were people who were just curmudgeons that said there's no way this could ever happen, and there were others who seemed to me had a conflict of interest saying, oh no, we need to have investment in this area. So I wrote the article, and a couple of years later, the World Health Organization published in 2018 a list of blueprint priority diseases. And you can see the 8 diseases, and that's probably how most people read this list. It's not the way I read the list. I saw one thing I was really interested in. Nipah made the list of blueprint priority diseases. And then when the Coalition for Epidemic Preparedness and Innovations stepped forward to develop vaccines against emerging threats, they initially targeted Lassa fever, MERS, and Nipah, working to move vaccine candidates to human testing, at least through phase 2, and manufacturing and stockpiling. So there are 4 ways a Nipah vaccine could help protect. The stockpile of an effective vaccine reduces the attraction of Nipah as a synthetic pathogen. It allows us to immunize health workers in endemic areas and researchers. It could be deployed in an outbreak of a highly transmissible variant, and prophylaxis of populations at increased risk for exposure. So you could imagine if the second measles vaccine across Bangladesh was a combined measles Nipah vaccine, and that's one of the vaccine constructs, then you could imagine not only protecting people against Nipah, but also protecting people against measles, but they'd also be protected against Nipah. And therefore, even if they drank contaminated date palm sap, they may not develop infection and disease, and so that it would not be passed on. So in conclusion, Nipah virus is a bat virus that infects people each year. It has a high case fatality rate of 75%, and it can be transmitted from person to person. Careful local investigation improved local response capacity and actually then went on to inform global action. I'm happy to take questions.
Thank you so much. My bad. Forgot about that. Anyway, so yeah, if anyone wants to unmute themselves and ask their question, they can, or they can put it in the chat and then I'll be happy to read it off to Dr. Luby.
Dr. Luby, maybe I'll start. Thank you. It was really interesting. My name is Rabin. I'm a former Stanfordite and been at Emory for 10, 12 years now. So I'm definitely becoming a Hawks fan, but I'm still a Warriors fan too. So I guess both of them are low impact at this point. Question regarding, so I'm an immunologist, and I'm really interested in the pathogenesis and whether you think, in addition to maybe antivirals and vaccines, maybe if there are if the high fatality rate is caused in part by maybe an autoinflammatory syndrome in the patients, and whether you think there's any space there for maybe host-directed therapeutics.
Yeah, I think there is almost, yes, I think we have evidence that there is a dysfunctional immunologic cascade. And one of the reasons that I say this is that if you just look at the severity of infection across mammals, it differs markedly. Pigs can get infected, but their case fatality rate is only about 5%. And they typically have a much more mild disease. And they mount antibodies, and they get, so there's, and it does look like this is just a negative cycle that just keeps getting reinforced when these patients start to deteriorate. One of the challenges is that across South Asia, these cases tend to come to attention late in illness, and tend to predominate in rural areas, not in your leading medical centers in urban areas. So they often don't get recognized early either. So I think there are some particular challenges in terms of interrupting the pathophysiologic process that late in the game. But I also think that given the severity of this virus, anything that we can learn about how to modify the immunologic process is something that also could generalize to other potential pathogens. So I see it as an area writ large that's worth giving more thought to.
Yeah, as a quick follow-up, I was really thinking about the bioterrorism threat potentially. And the other one was, as a quick corollary to the question, do you see a clear age-dependent fatality as well? Do you see, protection of the little ones and, more fatality in older people?
It is not strong. 75% case fatality, and we see kids as young as 3 years old dying. So
but below 3, you don't necessarily see that because they don't consume, or because they don't die?
Because they don't consume.
They don't consume. Okay.
Yeah.
Because for SARS-CoV-2, we see a similar thing, and part of what we're thinking is, at this point as immunologists, is that there's a window of opportunity where the body has a more accepting innate response as it's building memory against exposures, and then once you build that memory, you switch to a more antibody dependent type of responses where your innate is basically poised to respond to things that are bound to that are brought to it via antibodies, right? So you kind of using your adaptive to trigger your innate. Well, in the beginning, you're using your innate to inform your adaptive, right? And that switch might be explaining in part the difference in fatality for SARS-CoV-2 in particular. So I was maybe expecting something different, but consumption definitely makes sense as well, that the little ones would not be exposed to that. So thank you.
And I think that one of the things that, as I reflect on with emerging infections, is that just different viruses are different. We spent so much time thinking about the risk of pandemic influenza in Bangladesh that when SARS-CoV-2 came up, I kept thinking influenza, but the coronaviruses are different, and it just took a while mentally to make that switch. So yeah, I think there are some things that are peculiar about this virus, but I still think understanding and interrupting the immunologic cascade is still a high priority area.
Thank you.
Yeah, go ahead, Henry. Henry, ask your question, or you can put it in the chat and I can say it for you. We aren't hearing you if you're speaking.
Can you hear me now?
Yes, we can hear you.
Oh, sorry about that. Okay, sorry, speaker issues. Thanks for the epidemiologic investigation. I'm just really curious, what's the thought why drinking date palm sap for many generations didn't seem to cause problems in the past? Was it just an undetected illness, or is it something that emerged more recently?
Yeah, so they've been harvesting date palm sap in that part of the world for centuries. So, and they're using basically the same approach. And the bats have been there forever. No, the bats have been there for millions of years, yeah. So I suspect that these spillovers occur and were undetected. And the reason is that, again, this doesn't happen in cities. It happens in rural areas where people are harvesting the sap. And having some poor people die in poor communities is just not that uncommon. We were able to identify Nipah because we set up surveillance, because the outbreak in Malaysia had isolated the virus, and so given rise to diagnostics. So my hunch, and the other thing is that lots of people drink raw date palm sap, and it's low probability that you drank one that was infected by a bat that was shedding. When we capture bats and test them, it's usually less than 1% of them are shedding virus. I mean, we get some RNA, usually somewhere between 0.5 and 2.5% of them will be shedding. So you need a pretty unlikely arrangement of risks in order for this to be a transmission event. You need to have a bat that's a relatively heavy shedder coming into date palm sap that's not directed into making molasses, but this is the date palm sap that is drunk raw and that people get enough of a dose. So I think this probably went on. I think people were dying, but because it's sporadic and because the chains of transmission were never in a high density area. So I think that these would have been terminated. So yeah, I think it went on and we just didn't see it until we had the tools to look better.
Steve, I have a question. I really love the talk, and it's really great how you're connecting all these different skills, which is really nice for our program. Especially thinking about, you need to really understand, and as part of these local investigations, what is going on, to have any sense of what may happen ultimately at the global scale. So my question is a bit more about the bats, and just like looking at the map that you have up there now. So you showed the phylogenetic tree suggesting that these different clades of viruses separated about 75 years ago. And I just wondered how much is known about the bats and how the virus spreads across the bats, and how, are those bats, say in Bangladesh and Malaysia, are they connected somehow? Is that one big panmictic population? Is there migration of bats? How are these things connected?
Yeah, so those are great questions, and there have been a bunch of bat ecologists who have helped us think through all of this. So everything there in yellow, those are genus Pteropus bats, but there are different species. So the species in India and Bangladesh, Pteropus medius, are different from the Pteropus hypomelanus, and there are actually 4 other species going across Southeast Asia. Now, of course, they descended from each other, and they all seem to be able to carry the Nipah viruses just fine. If you look at the population of bats that have been collected across Bangladesh, there's one group that argues that there is a genuine population difference looking at the population biology of the bats. So we might think of them as being a couple clades of bats. And I think that we don't have enough sequence data for me to be persuaded anyway that that the Nipah virus has predilection towards one or the other. These bats tend to roost together in large roosts where they pee on each other and lick each other. And so there's a lot of fluid transmission there. And often even those roosts can be shared with other bats as well. And then if we step up not just to the genus level, but to the family level, Pteropopetidae, then you get even a broader group of fruit bats into Africa. And some of those bats, there have been isolated RNA from viruses that look like Nipah virus, but we don't know anything about human infection and human consequences of that. So my sense is that the Nipah virus in these bats co-evolved over at least thousands of years. And there probably are some bat population differences that drive some of this, but I think we don't yet know all the details to sort that out.
Thanks.
Go ahead, Anne.
Hi, thanks for the great talk. A broader question, just in general, like why are bats such good reservoir species for all of these viruses, and they don't get sick? And is there something about their immune system, or any reason why that might be the case?
Yeah, bats are remarkable, aren't they? So one thing is, so I think there are lots of reasons. I don't think there's one. One is there are a lot of bats. There are like 1,300 species of bats. They're like 40% of all mammals, or 30% of all mammals. So there's just a lot of bats. So they have lots of opportunities to be found time and again to be reservoir species. So I think that's one. Second is that bats travel far away. And especially the fruit bats. The fruit bats are seed dispersers. They'll fly even hundreds of kilometers when you put satellite trackers on them at times. So that if they are cases, so they're good at moving potential pathogens around. But I think the third issue that you have suggested is also really important. The bat immune system is just different. It is just very different in terms of the way, particularly it processes innate exposures. And I think that we don't know all of the differences, except to just say that when you put a variety of viruses into bats, they respond very differently than in other mammals. So, and I've read some argument that the bat immune system evolved differently because bats get caloric deficient when they fly all over. I don't know, that seems kind of hand wavy to me. But I do accept that there's a qualitative difference in how a variety of pathogens are managed by the immune system, especially the innate immune system in bats. And I suspect that that's one of the reasons why these viruses can persist, because this is a paramyxovirus, so the bats never clear the virus. And that's one of the reasons why they intermittently shed. It's a little bit like people with oral herpes. You never get over it. You get cold sores when you get stressed and all. So I think that's one of the reasons why this virus has been able to persist so long, is because of the long-term infection in the bats. Yeah, and the bats have an odd immune system.
Okay, thanks so much. We're actually going to wrap up here because we have Dr. Luby going into some meetings with some of us. So thank you all so much for joining this seminar. And thank you so much, Dr. Luby, for that wonderful presentation. It was super fascinating and I really, really enjoyed it. So yes, everyone give a round of applause.